Now we would look at the starch degradation that takes place in tubers, and as we know that this starch degradation has major impact on the quality of end product uh, in terms of the amount of sucrose or amount of simple sugars which are generated by the action of different enzymes on starches to produce simpler sugars, and that in turn would have effect on the quality, desirable attributes of these particular varieties of potato. And as we know that again this degradation, because different cultivars are different and they have got different starch chemistry as well as other attributes, so this is variable among different cultivars of tubers, that how starches are degraded in different cultivars. So if you look at this modification, this is basically a chemical modification that happens by the attachment of glucose with the phosphate esters. And these phosphate esters, they attach to C3 or C6 glucosyl residues of amylopectin. And this happens at the interbranch in the interbranch area, and not at the branching points. So this would actually, once they are attached, these esters are attached, they would make up a weaker area in the chemistry of these branches. So they can be cleaved at this stage. So that would mean these are the weaker area of this chain, that because we have had these phosphate ester, they are attached to these branches of the amylopectin, and once they are attached, they would make the starch more prone to degradation or breakup by the action of different enzymes. Another factor that is also equally important is the some of the negatively charged surfaces that do come from whether lipids or proteins or phosphate levels as well. They would also have great impact on the action of different enzymes that would be whether helping or stopping different enzymes to break down these starches in the tubers. So these two factors basically would define that how prone is a particular starch for action of different enzymes, and that in turn would lead to the breakdown of starches. So different cultivars would behave differently depending upon these two factors. And generally, this reaction takes place in amyloplast. The degradation of starch happens there in the amyloplast. And as we know that there are different concentrations of different enzymes such as alpha glucone pyrophosphorylase, alpha amylase, beta amylase, and maltase. They are all enzymes that are also present in the same area where the starch biodegradation is taking place. So this biodegradation can be through two means. It could be phosphorylytic, or it could be hydrolytic, or it can be both different two mechanism in which this degradation takes place. And this we can assume from this information which is available, that the concentration of these particular enzymes is more prevalent in this area where the degradation of starch takes place. So once we, as we earlier said, we transfer these potato tubers from the field crop to the chilling temperature for storage. The sucrose is a sugar that would start accumulating in these in the storage conditions. As we said that this is quite a consistent phenomena that is visible, and of course it's not desirable. And we also know that the activity of amylases or these enzymes is very low at low temperature, because the temperature is quite low, the storage temperature, and they would not be very much active at this temperature. So then we can assume from this that the starch breakdown that happens during the cold storage in different tubers, that would be mainly due to the phosphorylases. These enzymes would be more responsible, because the low temperature would discourage the amylase to bring about the biodegradation of these starches. This is again the type of potato phosphorylases that are used. They can be based on two criteria. For example, the specificity, glucan specificity, where they actually act. Then the monomer size, like how small they can react with how small molecules of monomers, especially the sugar monomers, or intracellular location, where these enzymes are located within the intracellular positions. So on that basis, they are basically, because they are enzymes, as you know, they are all proteins in nature. They are interconvertible. They cannot be interconvertible. They are non-interconvertible. Their different structures are different, and their properties are also, immunological properties are also different. So we know that there are two types of these enzymes. Type 1 is also called as type H or type 1. This is an isozyme that is present in cytoplasm, and it has got lower affinity for maltodextrin and higher affinity for branch polyglucans. So we know that it would be more affecting or reacting with the maltodextrin than polyglucans, and this would be cross-reacting with type H phosphorylase from the potato leaves. This is one enzyme, as we said, and the second isozyme is called as type L or type 2. In reverse to the previous one, this would have more high affinity for maltodextrin and low affinity for branched polyglucans. So these enzymes act differently, and it also cross-reacts with type L enzyme from the leaf. So these enzymes have got very important role to play. Another important element is the presence of inorganic phosphate that has got also a role to play in the degradation of starches. Normally, this intracellular compartmentalization of inorganic phosphate, it would be having impact on partitioning of non-photosynthetic potato tubers, especially the carbon partitioning that is due to these inorganic phosphate molecules. And they would be they start shifting their metabolic activities toward starch degradation than accumulation. So they are more inclined to break down the starch rather than to synthesize starch. We also know that the effect of inorganic phosphate is also quite visible on ADP GPase enzyme that we earlier said. It is a rate limiting kind of enzyme present for biosynthesis of starches. So this indirectly is also influenced by inorganic phosphate compounds that are present within the affinity of the cytoplasm. So another source of inorganic phosphate is also, they are also present in the vacuole part of the potato tubers, and it is compartmentalized it is away from the cytoplasm. So this, again, the location of this inorganic phosphate and its effect on ADP GPase enzyme, that would be very much influencing the type of action of these enzymes, and that type of action of enzymes on these starches for degradation would in turn lead to the outcome, such as the breakdown of starches towards desirable or undesirable. That would depend upon that what is the type of the mechanism that is involved there. But these things are important to be known by biotechnologists, and these can be exploited to make the best use of this information to produce desirable products with desirable starch chemistry, and that starch chemistry, as we know, would have impact on the functionality of starches.